Welcome and thank you for attending my lecture. I will be covering the lecture on metabolic treatment of epilepsy and other brain diseases. I am a neuroscientist and I'm interested in molecular neuroscience as it relates to the operation of the brain and also on the physiology of the brain itself. I'm also a neurologist and I have expertise in unusual or undiagnosed diseases with particular interest in brain metabolic disease. I am grateful to NIH staff, particularly Adam Hartman, for conceiving the idea of the workshop and inviting me and creating most of the agenda. He's been greatly helped by Vicky Whitmore. And thankful to my co-chair, Mackenzie Zorenka, for being wonderful to work with. My laboratory members, because I'll be presenting results that they have produced. My institution for being a great place to work at, and then NINDS for funding us, really without interruption for quite a long time. And finally, and very especially to patients and families, because they have been the reason why we do what we do, and they have been quite helpful in a number of clinical research initiatives. I'll be covering 4 main topics. I will introduce what metabolism is. This is a very broad overview, and there is also a great diversity in the audience, so I will try to be fairly generic. I will then move on to justify why metabolic therapy is relevant or important, and the rationale is that most of them increase rather than decrease function, and also because they're quite versatile, they can be accomplished in several ways. I'll move on to discuss some of the principles by which they operate. They all support neuron excitation, but they also participate in non-metabolic processes, and those are pretty important as well. And finally, I will conclude with examples from the epilepsy field and the dementia field to illustrate the scope of these therapies. So what is metabolism? It's nothing but the import of matter into the body and the conversion of that matter into the energy and other molecules that have to be provided so that function is preserved. That's how much the brain consumes. It's not very much, but it's a very high intricate mechanism for procuring that constant energy supply or production. Now there are 2 sides to metabolism. One is the import of matter from the outside into the organism, represented as these logs taken out of a forest, and that becomes then the segue into 2 different aspects. There is a destructive aspect, here represented as a fire. This is a consumption of all these products, and there's also a constructive aspect, represented as that violin. And we call the source of metabolic substances, we call that fuel or prime substance. We call the destructive component catabolism, and we call the constructive component anabolism. Every process in the organism, it's always a balance of these 2 different aspects. Now, it's also important to keep in mind that metabolism, and particularly brain metabolism, is highly age-dependent or developmental dependent. This is the amount of glucose that the human brain consumes on a daily basis. So about 90 grams of glucose consumed by a normal brain. This is the adult level of consumption. And here is the age dependence of this glucose consumption rate, all the way from birth to age 15 or so. And initially there is a very low, relatively low glucose consumption rate in the newborn, and then that increases rapidly until about the age of 5, and it really exceeds brain glucose consumption in the adult, and eventually it will decline to adult level. Now that's made relative by taking a fraction, the fraction of that brain consumption divided by the rate of total body consumption represented in the bottom. And of course, because the denominator of the fraction is also changing, the result of the fraction, it's a different shape as the brain consumption alone will be. There's initially a decrease, then there is an increase, then a further decrease. And that's really relevant for individuals who are designing metabolic therapies and metabolic consequences, because you have to keep in mind what happens to the rest of the organism. Now, why the rationale? Why are these meaningful treatments? Why have they received attention and will continue to do so? Well, because they tend to augment rather than suppress a process. I'll give an example from a different field. Sometimes it's easier to see examples for non-metabolic areas. Here is a case of a child in Germany, about age 5, born very strong. He could hold that 7-pound weights in his arms. His muscles are twice the size of an adult, of a child his age, and he had less body fat. And that's because he had a mutation in a particular gene that allows the muscle to grow larger, and that gene is called myostatin. Here is a child at 7 months of age, and you can see that, yeah, his thighs are broader, as is the calf. It's a very strong child who has this mutation. Now that's relevant because it's actually possible to take advantage of this muscle variant to treat central nervous system disease. So here is another example from a different disorder, spinal muscular atrophy. This is a motor neuron disease represented here in a cross-section of the spinal cord. This is a primary site that degenerates in SMA, and then as a result, the muscle gets weaker. Now, it's possible, as I said, to compensate for this process by suppressing myostatin, therefore leading to enhancement in muscle performance, in muscle mass. That's quite relevant because here you're manipulating a component of the network that's far removed from the site of pathology. So treatment doesn't need to occur, doesn't need to proceed through the primary pathophysiological process. It can be a compensatory event relatively removed from the initial site of disease. And so keep in mind also that most of the drugs that are used inhibit. It's pretty unusual to come up with an activating drug in medicine. Most of them block something or prevent something. And as we know, inhibiting usually leads to significant side effects. It's also important that when something gets inhibited, it's relevant to comprehend as much as possible about the mechanism of inhibition and about the mechanism of the side effects. Now, that's how drugs operate. Metabolic therapies are very different. They usually increase or augment physiological process that's already present. And so you are taking advantage to some degree of a natural process that's simulated. And so it's not as important to comprehend the full disease mechanism if you're going to be augmenting something that is not directly the disease mechanism. And so they're also quite versatile. And by that, I mean that there are different ways to achieve often the same metabolic result, because there is some redundancy in many of the metabolic avenues, metabolic pathways, biochemical reactions that underlie metabolism. And that's represented in here. Anybody who's seen this biochemical chart knows that for many of these products, there is usually more than one way to achieve the same result. And in real life, we know that if we are late in taking a particular route, we can always take a detour. We get to the same place, it may be a different avenue, but eventually you can get there if you take an alternative approach. Now, how do these therapies operate? Well, for one thing, they are crucial in maintaining brain excitation, and that's obvious. I will go into that in some detail, but it's also pretty clear that they also participate in non-metabolic or non-canonical processes, which are pretty important as well. This is an example when the ketogenic diet was being developed. It had been known since the 19th century that individuals with epilepsy, when they fast or they get malnourished, they improve in terms of the seizure severity and seizure frequency. And this is a report from 1921 where an observation made at my former institution at Columbia University, where a number of people with epilepsy were subject to severe fasting, improving their epilepsy, were then the basis for considering not fasting them, but actually switching their fat ratio in the diet, and therefore achieving a similar level of ketosis, ketone body production. Here is one such example, which led to the very well-known ketogenic diet approach that's used throughout the world today. Now, it's also important to keep in mind that very early on, it was identified that cell-to-cell communication in the brain is crucial, and that happens to be supported by primarily an electrical process, but also underneath there is a metabolic process. They cannot be the electrical event without supporting it with the metabolic event. And the quantitative demonstration and the rationale really crystallized in the 1930s and 40s through the work of Andrew Huxley and Alan Hodgkin, and they work on invertebrates, and this is one such animal, the squid. Squid has a particularly large axons that go into the mantle that help with the construction of this large muscle. And we are going to be zooming into one of the axons. Follow the green square over the next few slides. Here is a section of the axon magnifying what I showed you a moment ago, and here is this single cell axon crossing the mantle, and this is a pipette inserted into the axon itself. They're very large, they're 1 millimeter in diameter, so they're relatively easy to work with. And here is the resulting electrical impulse. That is nothing but the flow of sodium and potassium ions in this one case. This is electrical current arising as a result of ion movement. But they also realized very quickly and very early on that in order to replace these ions that had moved, you need the operation of a pump or a different constituent that will replace, restore the ions where they belong. Otherwise, excitation would be followed very quickly by silence. The ions have to be put back. And so they knew that you need this is a section of the axon membrane represented in a larger cross-section. Ion channels will govern the flow of these ions that I showed you a moment ago. But there's also a pump that will restore the equilibrium, and that pump happens to be ATP consuming. There cannot be any operation of the pump without significant metabolic production of ATP, and we call the ionic part of the system excitation, and we call the ATP producing part of the impulse system metabolism. Again, there cannot be one without the other. There can be plenty of metabolism without excitation. That happens in many organs, but there cannot be any excitation without metabolism. And I would just highlight the importance that these are two sides of the similar process. You cannot have one without the other. And so what's relevant also is that this metabolic support of excitation is a fairly cell-specific or fairly region-specific. These are uniformly important processes, but the nature of that importance is different based on the cell type, based on the region of the brain or the core, and so on. And here is a cross-section of a brain representing the thalamus, which is a structure deep within the brain tissue, and the thalamus is connected with the surface, the cerebral cortex. These are excitatory connections represented as a plus sign, and there are two particular cell types in the cartoon here. The triangle is a cell that usually excites, and the sphere in green, it's a cell that usually inhibits. So excitation will come in from the thalamus, and both cells will be activated. This one continues on to excite something else. This one actually will, upon activation, will inhibit the other one, therefore terminating the activity in the circuit. Now, this happens to be a very rapidly firing cell. It's the one of the fastest firing cells in the organism. We call them fast spiking neurons. And here is an example of what happens with a very specific metabolic defect, pyruvate dehydrogenase deficiency. This is an enzyme that's a gateway to the Krebs cycle. When that's deficient, this is a mouse with pyruvate dehydrogenase deficiency throughout the brain. The firing frequency represented with this plot, it's far less than normal firing frequency represented in blue. So less firing for this cell. Now that's relevant because that happens to be a highly selective phenomenon only on that cell, not on the other. The other one is firing okay by itself. Here's an example of a widespread enzymatic metabolic defect that's quite selective within that part of the network or the circuit. In red, I will be showing data from our lab. Now that's relevant because in the in vivo situation, that has repercussions for how the brain works. This is the same mouse model, and here is the fidelity of the excitatory big cell that I showed you, followed by the firing of the inhibitory 5-spacking cell that I showed you in this network. They're very closely aligned in time, very close to zero, the firing of one relative to the other. In the disease model, in the pyruvate dehydrogenase enzyme defect, there's a lot more dispersion in the firing of one cell followed by the firing of the other. So the cortex of the animal is exposed to a much more dispersed barrage of action potentials as a consequence of these metabolic defects. So pretty significant network or circuit impact from one metabolic defect that predominantly impacts one cell type within the brain. Now, I also mentioned that there are important processes in that they're involved in non-metabolic phenomena, and such as regulating other things, cell migration, cell invasion, cell death, and so on. And I'll present a different example, again, from a different area, where a defect in an ion channel, that it's only engaged in allowing ions to move, actually is related to malformation or dysgenesis. How does an ion channel lead to a malformative syndrome? Well, because there are regulatory phenomena that are linked to that particular ion channel presence and operation as well. Metabolic processes are not any different. And I'll put another example up here. This is a well-known metabolic disease, acid maltase deficiency, Pompe disease. It's one cause of neonatal hypotonia. Here's an infant showing the hypotonia. In fact, it's said to be a muscle disease. Normally, children with acid maltase deficiency, if anything, suffer from motor neuron disease before they have significant muscle disease, but let it be. And what can be shown is that even though there is a glycogen built up in this defect in muscle and in liver, there is a widespread metabolic disarray within the blood representing other components. The details are not important, but different constituents of signaling have been altering blood, represented in gray here. And if we analyze some of these metabolic aspects, it's possible to correlate this dysfunction with dysregulation. And the bottom line from this sort of work is that in this one example, likely there is a dysregulation of a very important regulatory protein called AMP kinase. So again, a very focal disorder causing glycogen deposition, causing an array of problems, and yet having a very wide, a very broad regulatory consequence. Here is another example, unsuspected aspects to metabolic defects. In this one case, again, I'll show what happens with the pyruvate dehydrogenase deficient mouse. It is that they have a very permeable blood-brain barrier, and that wouldn't be extracted or assumed from first principles by knowing what metabolism is. So here is a normal mouse on the top left, and then on the bottom, he has been given a blue dye. It's harmless and it goes away after a period of time, but the tissues of the mouse acquire blue color just because it penetrates everywhere. It shouldn't penetrate the brain. The blood-brain barrier will keep it unchecked, so it doesn't get into the brain tissue. And on the left here is a normal mouse that had been given the dye, and there is no blue in the brain. Here is a pyruvate dehydrogenase mouse. It's a smaller brain because of the disorder, but it's also blue to some degree just because the blood-brain barrier has been open or breached or not formed. And that will have very likely very significant consequences for the development of the animal and for the function of the animal. So here's a non-metabolic example of primary metabolic enzyme manipulation. Another example, it's also possible to correlate metabolic activity with things like cell invasion in cancer. this is work done in glioblastoma, where the subject on the top left has significant glioblastoma in the temporal lobe right here in the MRI, and this is a PET scan showing a great deal of glucose accumulation within the glioblastoma, and these are the cells that came out of that tumor. Now, it's possible to infuse carbon-13 label metabolic substrate, in this case glucose, into the person, and upon tumor extraction perform nuclear magnetic resonance spectroscopy, NMR, which is shown in here. And the highlight is that there's a great deal of metabolic activity within the tumor past glycolysis. So it wasn't really thought to be, tumors were not thought to be very mitochondrial dependent in terms of metabolism. We were able to show that they have a great deal of robust mitochondrial metabolism, and these happen to be atoms within neurotransmitter molecules like glutamate, glutamine, this is lactate, and so forth. Now, it's possible to take advantage of this to perform non-invasive MR spectroscopy in the unmodified brain, the individual who hasn't been given anything. Here's another example of an oligodendroglioma in that subject. Here is the lesion on this axial MRI section. And each of these voxels comes with a little proton MR spectrum. And represented in here is the presence of this metabolic byproduct, 2-HG, that's 2-hydroxyglutaric acid. Now, in the normal brain, which is here, it's from the same subject right here, there is no such thing. So it is possible to correlate tumor severity or tumor prognosis with the presence of metabolic enzyme mutations that will then allow this metabolite to emerge and to be identified non-invasively by MRI, and then try to prognosticate the outcome due to this primary metabolic abnormality. I'll conclude with two examples of these therapies. One will be epilepsy, the second one will be Alzheimer's disease. In epilepsy, it's very well known that seizures are sustained by metabolism, so this shouldn't be a surprise. And in dementia, it's also very clear that early metabolic impairment is one of the crucial phenomena well before there's other processes happening within the brain. This happens to be the brain glucose transport defect, type 1 deficiency on the left. This is a PET MRI of a subject. A normal individual will have a great deal more orange and more red, in fact, due to radioactive glucose accumulation. And here there's a great deal of blue and faint orange, represented low accumulation of radiolabeled glucose. And this region, particularly the thalamus, is actually unusually low in color, in intensity, low glucose accumulation in this defect. Typical EEG electroencephalogram showing a seizure. Every lead, every recording point in the scalp shows the same, about the same discharge. There is this spike and a wave and a spike and a wave and a spike and a wave, about 3.5 times per second. Now, here is how the disorder happens. These are section of the brain, blood vessel cut across. Glucose has to get out of the blood vessel and cross the brain tissue, getting into neurons. Represented in green, it's a synapse, it's a presynaptic element, postsynaptic element, synaptic vesicles, and neurons have GLU3. That's the glucose transport mechanism primarily. Everything else is GLU1 dependent. So from the blood vessel wall all the way through the astrocytes, intermediary cell types that are here intervening within the brain, every one of these steps is dependent on the GLU1 transporter. And so what happens here is also, again, a fairly selective excitation defect. Complicated slide, but these are synaptic events that are happening in a particular cell in the thalamus. Again, these are inhibitory cells, and this noisy line happens to represent electrical activity over a period of time, and each of these transient signals happens to be a synaptic event. This is how many of them there are, and the recording continues and goes on and on and on. In the mutant, GLUT1 deficient mouse, there are fewer of these synaptic events. That's in low glucose. Remember this glucose transfer effect. If we apply high glucose to this preparation, the frequency of the synaptic events is actually restored to normal. So represented in here. This is the frequency of these synaptic events. Normally you have this many. In the Newton, they're about 40% of normal. With high glucose, they are essentially normalized. The size of the events, when they happen, the amplitude, this electrical current size, is actually unchanged no matter what. It's just a frequency. And if you apply acetate, which is an alternative fuel, the frequency of the events also increases. So here is bypassing metabolic defect by alternative route, and also enhancing the deficiency by just increasing the substrate, not increasing anything else. And so one avenue, since you could conceivably bypass such a metabolic defect through an alternative route, was to apply this triglyceride to subjects with the deficiency. This is an ongoing clinical trial where we're giving triheptanoid to individuals on a normal diet and now also on a ketogenic diet itself. And this is nothing but a conjunction of glycerol with 3 7-carbon fatty acid molecules. That's heptanoic acid. 3 of them conjugated into the molecule. They get cleaved, taken up by the liver, and they produce 5-carbon ketone bodies, which then penetrate the brain, and they give rise to propionate coenzyme A and succinyl coenzyme A. And those happen to be very powerful stimulants of the Krebs cycle, leading to the production of glutamate and glutamine. And this is represented in here. This is a complicated slide also showing the metabolism of the 5-carbon ketone body arising from triheptanoid metabolism within the brain. And as I said, the reactions will proceed, and eventually this label, in this one case, carbons 3, 4, 5, in the heptanoid molecule will emerge, will appear in neurotransmitters, and that's represented in here in the yellow boxes. So this happens to be the glutamine NMR spectrum, and this happens to be the resonance that arises from carbons 2 and 3 in glutamine. And in the normal mouse, there is that much doublet production, and in the mutant mouse, is very significant increase in this production. So therefore, it can fuel brain metabolism effectively. And in subjects, this is what happens. This is one typical seizure event. Here is a seizure right here for the subject. Here's a brief pause, another one, another one. You can quantify the duration of the seizure event and divide it by everything else in the recording. And you can consume the triheptanoid product. And right before there is a frequency of seizure events, you consume it. And after about 25 minutes or so, the seizure frequency is represented here, relative to baseline seizure frequency. So we did this in a number of subjects, and here's the results. The panel on the left is the same panel on the right, which would just amplify the range here from 0 to 0.2 seizure frequency right here, just so this group here can be easily seen. But pretty much everybody had a reduction in this seizure and duration in comparison with baseline. The important thing is that it also helped with cognition, and that's crucial in neurology, where many treatments will treat the electrical phenomenon or some other biological phenomenon, which really doesn't matter very much to the subject. This is different because here we were aiming to look at cognition, and this happens to be one particular cognitive test, and this happens to be another. This is a baseline for a number of subjects, 1 dose, 6 months of treatment, and in some cases there is enhancement or improvement or increase in some of the cognitive measures, so therefore justifying looking at this in more detail. And finally, like I said, I'll give the example of Alzheimer's disease. It's been very well known actually for quite a long time that the very first thing that happens, in fact, it's a decrease in the amount of glucose that's taken up by the brain. These are these are PET scans. On the left, there is somebody with depression, top and the bottom. Here is somebody with multi-infarct dementia. That's a not infrequent condition. And the one on the left, it's near normal in terms of glucose distribution. The one on the right, wherever there's an infarct, there's a decrease in glucose accumulation, as would be reasonable to assume. But the one on the right, on the far right, has Alzheimer's disease. And very early on, there is this very significant decrease in the amount of radio-labeled glucose that made it into the brain. There is this parietal temporal cortex abnormality that's very visible, in addition to some frontal abnormalities, and these are very significant decreases. So there's been a great emphasis in treating Alzheimer's disease now with metabolic intervention. There's a variety of things that are being tried right now at different stages of development. This is just two of them. There is a peroxisomal proliferation agonist, and there's also a thymine derivative. And finally, there's also a tryptanoin as a bypassing that glucose metabolic defect. And this is work provided to me by my colleague, Hendu, who's been wonderful to work with. And he was nice enough to include me in this work, where he looked at this particular mouse that has amyloid deposition, the so-called 5X FAD transgenic mouse. They have amyloid plaque deposition, not too different from what a human being has when they have Alzheimer's. And here is a number of synapses in the hippocampus, this particular region called CA1. It's very important. It's a test bed for many interventions and also for brain physiology, where the number of synapses happens to be that many in the volume that's been analyzed, and the transgenic animal has these many fewer synapses in that region. With tryptanoin, nothing much happens to the normal mouse when they're being fed the diet. However, it fully prevents synaptic loss in the mutant mouse to resemble what it is in the normal mouse. So here is a representation of the synaptic sites, showing that this particular treatment is, in fact, promising. I conclude. Again, thank you very much to everyone for being so helpful, and thank you to the audience. The references are given here for anybody who wants to look into some more detail, and I appreciate your time. Thank you.